Hello, my dear friends and future doctors. I am Dr. V. Mahendra, Assistant Professor of Biochemistry, Islamia College, Waniambadi. I completed my doctorate degree in the field of molecular clinical toxicology, and I have 10 years of teaching experience in the field of biochemistry. And I have n number of publications with the citation index of Scopus, Web of Science, and everything. It is my demo video. My topic is DNA replication. DNA replication is a process by which a double stranded DNA molecule is copied to produce two identical DNA molecule. That means during DNA replication, DNA commonly in the form of double stranded DNA and anti-parallel. One is 3' to 5' direction, another is 5' to 3' direction. So during the DNA replication, the double-stranded DNA is opened in a particular site is called as origin of replication or replicon. That open done by the enzyme namely DNA helicase. Now the synthesized strand always to be 5' to 3' direction. So one in opposite, that means is a bidirectional replication. So the process by which the double stranded DNA is copied, that means the newly synthesized strand is formed from the parental strand. The parental strand acts as a template strand and the newly synthesized strand is called as daughter strand. The replication is an essential process because whenever the cell divides, the new daughter cell is formed. The daughter cell should or must contain the same level of genetic information as the parental cell. So such a way the DNA replication in which the daughter DNA are synthesized using parental DNA as a template. See that red color is a parental strand where the yellow color stands for newly synthesized daughter DNA. The transferring the genetic information to the next generation with high fidelity, that means without any error, without any mutations. The parental DNA is copied to the daughter DNA. That means the DNA is made up of adenine, guanine, cytosine and thymine, the four bases. So the adenine always pair with thymine, guanine always pair with cytosine. Such a way, during replication, the daughter DNA is synthesized by adding complementary nucleotide by the enzyme, namely DNA polymerase. The DNA polymerase having the high fidelity and having a repairing mechanism also. It does not make any error during adding the complementary base pair during replication. So types of DNA replication. Generally, there's having three different types: conservative replication, dispersive replication, and a semi-conservative replication. The conservative replication, a hypothetic form of replication in which the double-stranded DNA produces two daughter double-stranded DNA. One is like the original strand, another is produce a daughter DNA. So it's like a Xerox copying. It's called as conservative mode of replication, but there is no experimental proof for conservative replication and dispersive replication also. In the dispersive replication, in which the original DNA strand breaks up randomly and the synthesized strand is formed by adding complementary DNA, and again it reassociates to produce a double-stranded DNA with parental and daughter DNA. In this dispersive replication also there is no experimental proof. And final type is semi-conservative replication. In the semi-conservative replication, the parental strands are separates and it acts as a template. It produces a complementary daughter strand. One half of that DNA that contain a parental strand, another half of the DNA that contain a daughter strand. It is called a semi-conservative mode of replication. The base pairing allows each strand to serve as a template for new strand synthesis. Each strand of the double-stranded DNA served as a template. And new strand is half of the parent template and half of the newly synthesized. It has the experimental proof. Messelson and Stoll experiment in 1958 proves the replication is semi-conservative model. In this experiment, the E. coli, the bacteria, which grows in the medium containing N15 isotope. It is a heavy isotope. So, during the bacterial replication, during the DNA synthesis, it takes up a nitrogen source as N15 isotope. So, first generation it grows in the medium of N15. Again the same E. coli bacteria, it transferred to another medium which do not contain N15 but it contain N14 isotope. It is low isotope. So after the few generation, the bacteria is isolated and again the DNA is extracted and it's separated through cesium chloride density gradient centrifugation, where the results are shown like this. In the first generation, the DNA is isolated from the bacteria, is having only one band, that heavy band, heavy isotope of N15. The next generation, the DNA is isolated and it is separated in the cesium chloride density gradient centrifugation, which shows a band between N15 and N14. That means one strand of DNA that contain N15, another strand of the DNA that contain N14. It shows a semi-conservative mode of replication. Further generation also the same DNA is isolated from the bacteria and centrifuged. After we get the N14 and N15, a band and one more band is fully made up of N14. That means newly synthesized. So each and every generation there are a band in between N14 and N15 that shows a semi-conservative mode of replication. So each and every round of cycle there is a parental strand of N15 and newly synthesized strand of N14, which continues each and every round of replication. Now we move on to replication in prokaryotes. The prokaryote DNA replication is a process by which the prokaryotic organisms duplicates, that means copies its DNA into another copy that passed on a daughter cell or next generation. The replication is bidirectional. Already I told that it is a DNA double-stranded. It's an anti-parallel. One is 3'-to-5', another is 5'-to-3'. So the synthesis strand always to be 5'-to-3'. So if this strand acts as a template, it is 5'-to-3'. So synthesis strand always to be 5'-to-3'. It is to opposite. So such a way that is anti-parallel. The DNA replication shown bidirectional replication, and it originates at a single origin of a particular site is called as origin of replication or replicon. So the prokaryotic DNA replication, it's having three different stages. One is initiation, elongation, and termination. The rate of replication in E. coli is 1500 nucleotide per second. That means the synthesis of daughter stand by the DNA polymerase enzyme by adding the complementary base pair to the parental strand template by adding 1500 nucleotide per second. So it is so the replication of E. coli complete within 40 minutes. So first stage of the DNA replication in prokaryotes is initiation. The initiation begins at a specific site in the DNA. It is called as oric or replicon. It is a unit of a genome in which the replication starts. It is the point of a DNA that's open up to form a replication eye or replication bubble by the enzyme helicase. So this is the origin of replication oric. It is a specific site. That means the origin of site that contain 245 base pair, in which that contain two segments. One is 3 repeats of 13 base pair sequence. That means that nucleotide a 13 base pair with repeated 3 times the same nucleotide sequence, and also that contain 4 repeats of 9 base pair sequence. That means a 9 base pair sequence which is repeated on 4 times. So this is the site of origin of replication in prokaryotes. It having 245 base pair, 3 repeats of 13 base pair and 4 repeats of 9 base pair. So this initiation, it starts by recognition of the origin of replication oric, and next the recognized DNA A protein, that means helicase protein, recognized at a particular site in the origin of replication, specifically 4 repeats of 9 base pair sequence. In this oric to form a complex of negatively supercoiled oric DNA wrapped around central core of DNA A protein. So first the DNA A protein recognize the origin of replication and it's bind the 4 repeats of 9 base pair sequences. It requires the process of histone like HU or 1HC protein that facilitate the binding of DNA A protein at 9 base pair repeats. Once the 4 repeats of 9 base pair is occupied by 20 to 40 additional DNA A protein. First, only one DNA A protein recognizes and binds on the 4 repeats of 9 base pair. Then 20 to 40 DNA A protein monomers bound on that 4 repeats of 9 base pair sequence in the oric. That resulting complex resembles nucleosome of a DNA, that means histone association, which negatively supercoiled oric DNA wrapped around the DNA core. That means these 20 to 30 DNA A protein wrapped around the 9 base pair repeats to form a nucleosome like structure. Then the histone like protein prevents non-specific initiation. That means the DNA A protein only recognizes the origin of replication oric and specifically bound on 4 repeats of 9 base pair. From other sites, the histone like protein prevents the binding of DNA A protein to other than the origin of replication site. The DNA A protein subunits then successfully melt. That means DNA A protein bound on 4 repeats of 9 base pairs, and it makes break up the hydrogen bond between the nucleotides in the 3 repeats of 13 base pair complex. So DNA A protein subunit then successfully melts the 3 tandemly repeated 13 base pair segment in the presence of ATP as an energy source, which results the formation of 45 base pair open complex. So first it binds on 4 repeats of 9 base pair, and it breaks the hydrogen bond and form a open complex on 3 repeats of 13 base pair to form a 45 base pair open complex that is called as replication bubble or replication eye. The DNA protein then guides, that means it recruits other helicase protein, namely DNA B protein and DNA C protein, a complex form of protein that melted to form called pre-priming complex. So DNA A protein, DNA B protein, DNA C protein comes in the open complex and makes continuously melting the DNA toward the helicase site. The DNA gyrase and a single stranded binding protein play important role during that open complex formation. If here the helicase makes the open complex, it makes a truss on other side of the DNA that can be relaxed by the enzyme called DNA gyrase or DNA topoisomerase. So that play important role to break one strand and releases the truss that created by the helicase activity and reseal that nick by the DNA ligase. So the single stranded binding protein bounds on the open complex and it prevents the re-annealing of the open complex formed by the helicase. So it is a hydrogen bond, it can be easily reassociated. So the single stranded binding protein comes to bind on the single stranded DNA and prevents re-annealing for the DNA. In the presence of gyrase and single stranded binding protein, helicases furtherly unbinds the DNA in the both direction. Here the DNA helicase, this here the DNA helicase, it makes to opening furtherly to make a open complex where it recruits other enzyme, namely primase or RNA polymerase and DNA polymerase. See here, the single strand binding protein prevents the re-annealing. Helicase, it continuously forms an open complex by breaking the hydrogen bond. Now the initiation process is over. Then the elongation process started. Elongation process started by the enzyme, namely primase or RNA polymerase. So we can see later. A replisome. It is the multi-protein structure that assembles at the bacterial replication fork or replication bubble or replication eye to undertake the synthesis of DNA through the process of elongation. So during that open complex form, it recruits other enzymes and proteins, namely DNA polymerase, RNA polymerase, primase, primosome, helicase, SSB, DNA gyrase, DNA ligase, topoisomerase and rep protein, all are assembled to form a complex is called as replisome. So the step is helicase, it unbinds the DNA, and SSB, it prevents the re-annealing, DNA kinases relax the truss. So the elongation process is started by RNA primase. This RNA primase or RNA polymerase, it synthesizes a complementary base pair of nearly 10 to 60 nucleotide to the parental strand. It is made up of RNA. That means it contains adenine, guanine, cytosine and uracil instead of thymine. So this RNA primer, it starts the elongation process by base pairing the parental strand. It is the parental strand. This primase synthesizes a complementary strand to the parental strand for few nucleotides. Now the elongation process starts. The elongation process starts by the synthesis of daughter strand continuously towards the 5' to 3' template is called as leading strand. During elongation, there is two different types of strands of synthesis. One is a leading strand, another is lagging strand. We are going to discuss why. See, already we discussed the blue color stands for double-stranded DNA, parental strand, it is template. It is the open complex is formed by the helicase. So here the helicase is there. The leading strand towards the helicase activity is continuously synthesize a daughter strand. That means here the opening is formed, here the opening that can done by the helicase activity. Towards the helicase activity, the DNA polymerase adds complementary base pair without any disturbance. So it continuously formed a daughter strand is called as leading strand. Here the lagging strand, it is opposite to the helicase activity. So there is a lagging is there. So there are so many primer required for synthesizing daughter DNA in discontinuous manner. It is called as lagging strand. So the leading strand synthesis begins with RNA primer. So the DNA polymerase, it's add a complementary base pair to the parental strand, but the DNA polymerase cannot start the replication by adding directly to the parental strand, but it needs some 3'-hydroxyl group of pre-existing nucleotide. So that can be given by the RNA primer or primase. So it adds few nucleotides in the base pair. The synthesis strand always to be 5' 3' direction. I said the synthesis strand always to be 5' to 3'. Here the parental strand is 3' to 5'. So here the RNA primer, it gives a complementary base pair to the parental strand for nearly 10 to 60 nucleotide. Then the DNA polymerase comes. It adds complementary base pair to the parental strand. The DNA polymerase 3 synthesizes the DNA by adding 5' phosphate group to the 3' hydroxyl group which is previously existed by the RNA primer. So it continuously synthesizes the DNA strand towards the helicase activity to the 5' to 3' direction is very fast done by the DNA polymerase 3. The enzyme is much more active than the DNA polymerase 1. That means prokaryotes. In prokaryotes, there are three different types of DNA polymerase is there. DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. DNA polymerase 3 is highly processed, highly sensitivity, highly specificity, and it adds very faster than the DNA polymerase 1 and 2. That means it can add 900 nucleotides per minute at 37 degree than the DNA polymerase 1. After it synthesizes the complete complementary bases, the RNA primer should be removed in the leading strand. That can be done by the enzyme called as RNase, because the RNA primer is made up of RNA, adenine, guanine, cytosine, uracil instead of thymine. But the DNA is made up of adenine, guanine, cytosine and thymine. So that RNA primer should be removed. That's done by RNase enzyme. So such a way, this is a parental strand, it is 3' to 5'. It is a daughter strand, it is to be 5' to be 3'. So DNA polymerase 3 adds complementary base pair. For example, guanine to cytosine, thymine to adenine, guanine to cytosine, adenine to thymine, thymine to adenine. Such a way it adds a complementary base pair to the parental strand. So the DNA polymerase adds continuously it synthesizes, that is called as growing replication 4 towards the helicase activity. In the lagging strand it discontinues because it's opposite to the helicase activity. Here the number of primer is much more. Here only one primer for leading strand synthesis, that synthesis by RNA primase or RNA polymerase. Here the primer synthesized by primosome, a complex form of primase activity. It releases continuously different primers, so it synthesizes discontinuously. That is called as lagging strand. That is experimentally proved by Okazaki, a scientist. So that fragment is called as Okazaki fragment. And also finally the RNA primer is removed by DNA polymerase 1. Here in the during leading strand synthesis, the RNA primer removed by RNase, but in the lagging strand synthesis the RNA primer is removed by DNA polymerase 1 by its 5' to 3' exonuclease activity. The DNA polymerase having polymerization activity, 5' to 3' exonuclease activity and 3' to 5' exonuclease activity. Here 5' to 3' exonuclease activity, that means it removes the primer. If it is 3' to 5' exonuclease activity means it is proofreading. That means after the complete synthesis of DNA, the DNA polymerase 1 comes back and check whether the DNA polymerase 3 correctly added complementary base pair. So A to T, G to C, C to G, T to A. Such a way the 3' to 5' exonuclease activity, that means a proofreading activity, comes to check whether it adds exactly complementary base pair to the parental strand. So such a way the RNA primer added by the primase in the DNA leading strand, primosome in the lagging strand. So here the continuous synthesis, here the discontinuous synthesis. The RNA primers are removed and the DNA polymerase 1 comes and it fill up the gap of primer removal, and finally the nick is sealed by the DNA ligase. So this is the lagging strand, it having different steps: synthesis of Okazaki fragment to the RNA primer synthesis by primosome in the lagging strand. It synthesizes 1000 to 2000 nucleotide are added by the DNA polymerase 3. In both leading strand and lagging strand, DNA polymerase 3 adds a complementary base pair to the parental strand. In leading strand is continuous strand, lagging strand is discontinuous strand. It is called as Okazaki fragment. That is due to leading strand it moves on the helicase. In lagging strand it is opposite to helicase activity. That is the reason. Excision of RNA primer. So after the synthesis of the daughter DNA by DNA polymerase 3, the RNA primer should be removed because it's made up of RNA molecule. That can done by DNA polymerase 1 by its 5' to 3' exonuclease activity. That means primer removal activity. Filling the gap. After the primer removal, the primer gap, that means primer is 10 to 60 nucleotide. So the primer gap is filled by DNA polymerase 1 by its polymerization activity. So DNA polymerase 1 having polymerase activity and 5' to 3' exonuclease activity. The joining of Okazaki fragment. Finally, the DNA polymerase adds only a complementary base pairs, but it does not seal the nick. Nick means phosphodiester linkage. This is the bond of hydrogen bond, the red color indicates. Here that strand is made of a phosphodiester bond. In between the DNA of daughter DNA and parental DNA, the bond is formed is hydrogen bond. But in between the nucleotides, there is a phosphodiester bond is there. So the phosphodiester bond is created by the enzyme called DNA ligase by the coenzyme activity, namely nicotinamide adenine dinucleotide. So such a way that daughter DNA is continuously synthesized towards the end by the leading strand and lagging strand. Finally, we move on to termination. During termination, there is a sequence is called as termination site or termination sequence is there. The termination sequence that contain the termination sites, namely A, B, C, D, E and F. Here the ter sequences, it moves clockwise by A, D and B. The ter sequence move anti-clockwise is done by ter B, ter F and ter G. When it meets the two ends, there is the origin of replication. The replication fork is moved like that and it reaches the end. When it meets, the termination is formed. So during that meeting, there is a protein, namely termination utilization substances, TUS. A protein is there, so it stops the helicase activity. So the helicase is breaking the hydrogen bond between the double stranded DNA. So here when it is reaches the clockwise and anti-clockwise movement of the ter sequence and TUS protein, when it is bound to the helicase, there is a termination is occurs. So when it meets a ter protein and TUS protein, termination utilization protein, the termination of the DNA, it stops. So finally the double stranded DNA acts as a template and it synthesizes another double stranded DNA molecule, a daughter DNA molecule, and a daughter cell is formed by which having the same amount of DNA exists as such in the parental strand. Thank you, my dear participant and future doctors. If you like this video, kindly support us. Thank you.